afternoon, everybody. We will start the webinar proper at 2:35 while we allow for more people to stream in. So in the meantime, do take a look at the webinar tips on the screen. Welcome everyone to the introduction of RIC supported technology platform webinar series. So I'm Paul from A-Star Research Support Center, RIC, and today we have Anna from Singaporean here with us. So today they'll be presenting to you on the topic of SynEcoSys, a user-friendly platform for single-cell data analysis, data mining, and visualization. So thank you for joining us today. So before I pass the time to our speaker, let me briefly introduce RIC to you. So RSC actually works in 3 key models for consumables and scientific services. So referring to the diagram, firstly, we provide a wide range of research consumables on our web store via the search click buy mode, which is the orange line you see on the screen. So besides consumables, we also offer scientific services from a variety of technology platforms available on RSC via the 2 other modes, the search book use, which is the blue line, and the Enquire Scope service, which is the green line. So we serve not just to A-star community, but these service offerings are also extended to the academia and private industry sectors as well. So you search book use, users can look for equipment they require and book the usage of this equipment, which can either be self-use or staff-operated. So while the Enquire Scope service mode is more specific and targeted, in which the relevant technology platform will carry out the service based on your experimental requirements provided. So we also have a broad range of services that we manage by the RSC technology platform, and we provide both the front and back-end support to this platform. So this allows them to focus more on the service delivery. So here's a brief overview of the services that we support, and it comes from within A-star facilities and also private companies that RSC partner with to meet your research needs. So RSC also supports 4 national shared infrastructure platforms. They are the recipients of the NRF Shared Infrastructure Support Grant with the aim to support researchers nationwide and in hope to advance Singapore's scientific contributions. So these platforms are available to researchers not beyond the A-star community. So here are the 4 platforms. The first one is the immune monitoring platform, which is based in SIGN, focusing on flow cytometry, multiplex soluble protein assay, and immunoinformatics. So the second one is Translational Pathology Consortium, which is based within IMCB. So they partner with various pathology facilities across Singapore, focusing on the in vitro and in vivo toxicology, as well as the histopathology services. The third platform is SynEcoSys, where it brings together the microscopy facilities from A-star, NUS, NTU, and SingHealth, making them available to the research community. And the last platform is SynMass, so which brings together the mass spectrometry facilities from A-star and NUS to the local community. So ASAF, which is also A-star Scientific Equipment Finder, is actually a search directory built on ARIN search technology. So the purpose is actually to provide greater visibility and access for scientists and researchers from the R&D ecosystem to the scientific equipment and relevant experts available within A-star. So all you have to do is to simply key in the equipment name or the equipment capabilities, and the search result will actually reflect the relevant equipment or facilities providing the equipment. So if you wish to find out more about ASAF, you can click on the link, you can via through the link or the QR code on the screen at the bottom right. So here are actually our upcoming events, and we will provide you with more information about these talks very soon. So do keep a lookout on our EDM. And if you have not subscribed to our EDM, our RSC newsletter, you can do so by visiting our website and provide us with your email address. So lastly, here is the QR code for the post-webinar feedback. It would be great if you can share with us what you feel about today's session and also what you'd like to see in future webinars. So I'll also send the link shortly in the chat. You can click on the link to get to the feedback form as well. So if you have any questions regarding RSC services about today's webinar, please feel free to reach out to us. So now I shall hand over the time to Anna. So do give us a few moments for the switch.
Good afternoon, everyone. So as Pearl said, my name is Anna, and I'm a product manager at Singaron for bioinformatics solutions. So today I will introduce you to our database solution, SynEcoSys. So SynEcoSys is a user-friendly platform for single-cell data analysis, mining, and visualization. As you know, single-cell sequencing is a fast-growing field, and as such, it has generated a large amount of data. The problem with having a lot of data sets publicly available is that in a fast-growing field, there are no gold standards data analysis, which means that we have a lot of data that has significantly differences in data analysis, being generated from different platforms, and it goes through different QC standards. Usually also requires experienced bioinformaticians to analyze this data, and it can always be subject to biases from the researchers, which makes it difficult to compare data sets from different research groups. So our solution to target these problems is to develop our own database. So Single-RON is a single-cell RNA-seq company that offers solutions that cover the entire workflow of single-cell, so from tissue to data analysis, and today I will focus on the bioinformatic analysis part. So very generally, there's two steps to single-cell data analysis. The first one is pre-processing. So you input your raw data from the sequencer, and you want to obtain an gene expression matrix. So in here, you have your cells, and you want to know how many transcripts you have for each gene in every cell. And these solutions usually also output a small data quality report. And in Single-RON, we offer Celloscope for this that can be freely downloaded at our GitHub page. So after you have your gene expression matrix, you can start mining the data and trying to understand and interrogate it to uncover biological insight. So SynEcoSys starts by automatically annotating your cell types and subtypes. And after that, you can do differential gene expression analysis, trajectory analysis, pathway enrichment. And I'll go into more detail later. But how? Moreover, SynEcoSys is a database where we focus on clinical samples. So we cover mostly human samples, and we try to cover as many disease states as possible. And I'll introduce to you how you can interrogate the different clinical metadata that is available. So let's try to understand how SynEcoSys fixes the problems that I mentioned before. So first, we have a high variability in data analysis. So what we do is that we standardize a method of data collection for public data sets, and then every single data set goes through the same pipeline that we call cell lines, meaning that the same parameters, quality control standards are applied to every data set. And also that the same cell type annotation is employed, meaning that it's easy to compare the data sets between themselves. Our cell type annotation is quite good because we have a lot of experience sequencing single cells, and we're able to automatically annotate over 2,000 cell types for human, mouse, and non-human primates. When you compare our data cell type annotation with other methods, it is quite accurate, either matching one-to-one with the publication or being able to distinguish cell subtypes from cell types. Another important characteristic from our cell type annotation is, as you probably, if you read single cell publications, you probably noticed that the names that the authors give to different cell types might vary between publications, but in SynEcoSys, we make sure to match the cell ontology, again, making it easier to compare data sets from different publications. So cell data analysis is not just for public data sets. You can also use SynEcoSys to analyze your own data. All you have to do is to upload your gene expression matrix. It will be run through the same automated pipeline, and your cell types will be automatically annotated. Of course, these labels can then be customized to your preference. Using the same automated pipeline also allows you to compare your data set with public available data in an easy click-through form. The next problem is that usually require experienced bioinformaticians to analyze the data. But in SynEcoSys, we collect data-wise statistics, so it's very easy to navigate through gene and cell type distributions, understand what information and where the genes expressed across different tissues and diseases. And we also have a graphical tool analysis tool called CellViz, where you can very easily click through and compare the different groups. So the data analysis visualization that we offer so far covers all of the single-cell RNA-seq analysis, from differential gene expression between the different clusters or cell types, between different conditions. So you can compare the expression of genes between patients that responded to treatment versus patients that didn't. You can visualize gene expression in plots such as this one here. So every dot represents one cell, and the darker the color, the higher the expression level. We also have cell composition, pathway enrichment, and cell interactions. And I will show you how you can explore these different data analysis and visualizations in our live demo. Moreover, as I mentioned, we are focused on clinical samples. So we collect every available clinical information for all the samples available in SynEcoSys. And we manually curated these clinical samples to allow for direct comparison across publications. So make sure that we use the same terms, allowing you for comparing different disease stages, comorbidities, ethnicity, all of this can be explored. So I want to show you an example. This, what you're seeing here is just a print screen of the metadata available for one of our data sets. So you can identify the patient ID, the disease, the stage of the disease, the treatment, which kind of treatment, ethnicity. So just by clicking on these boxes, you can select the groups you want to compare. And these can be important to find clinical biomarkers or potentially drug targets. But the best way to illustrate how easy it is to navigate and what insights you can gain from SynEcoSys is to use an example. And I would like to use the CD24 cyclic-10 immunotarget as today's example. So this Nature Communications publication generated single-cell RNA-seq for breast cancer for 6 patients. The next publication that I'm showing here from Nature took this data set and analyzed it and uncovered a potential target for cancer immunotherapy. So very briefly, the authors show that the cancer cells express CD24, and this interacts with cyclic-10 in the tumor-associated macrophages, preventing the macrophages from clearing the tumor. When the authors use an antibody to block CD24, this interaction is stopped and the macrophages are able now to clear the tumor. So it's clear to see as well that the expression of CD24 was restricted to the cancer cells, while the expression of cyclic-10 was restricted to macrophages. So this indicates that it is a potential good immunotherapy target, as it doesn't seem to affect other cell types. But the problem with this comparison is that, first of all, it only targets one breast cancer subtype, TNBC, and the data is only from 6 patients. So I wanted to see if I could verify these findings using a larger cohort. So in SynEcoSys, we have a larger breast cancer cohort that consists of 270 samples, and it is made up of several breast cancer subtypes. So I asked in which cell types is CD24 being expressed, and you can see, even though it is still expressed in cancer cells, we can also see it being expressed in other cell types, such as epithelial cells of the mammary gland. So this indicates that if we block CD24, we might be interfering with the normal function of healthy cells. But why do we see this difference? So in the first data set, we could easily verify that CD24 expression was restricted to cancer cells. But why is it different in this data set? So one option is that this finding is only true for this specific subtype of breast cancer. So when I look at the expression CD24 only in these samples, I again see that the expression of CD24 is not exclusive to cancer cells, but also found in epithelial cells of mammary glands and basal cells. So this is not the reason the author saw that difference. But we can keep exploring the multiple layers of metadata of SynEcoSys to try to find why we see this difference. Another option is again focusing in this subtype of breast cancer, compare patients that were not treated versus patients post-treatment. And in here, we can see that after treatment, the expression of CD24 becomes restricted to cancer cells. And this might be one of the factors for which you see these differences between the different cohorts. So it's always better to be able to verify your in silico targets in larger cohorts, because this will not only save you time from running validation experiments, but also costs. And you can do this type of verifications using SynEcoSys in 10 to 20 minutes. So next, I want to show you actually how you can generate this data and by showing you a live demo. So if you give me a second, I will share my screen. So in here, this is the home page for SynEcoSys. And SynEcoSys can be explored using 4 different ways. So you can look through tissue atlas, genes, cells, and study. And today I want to show you just a couple of them. So if you start by exploring the tissue atlas that you see here in the home page, you can click it. And we have one for human, mouse, and non-human primates. And then you have all the different systems that you can navigate by clicking on the cartoon here or in the side. And for each subtype of tissue, you can click on cell types and then expand every cell type, and you get to see the canonical markers that are used to annotate these cells. If you can go back and click on the system and then on tissue, it will take you to all the publications that are larynx samples. So it's very easy to navigate and find your samples of interest. Going back, you can also look for genes. For example, you type the name of a gene, CD4, and then you select your species of interest. I'm going to search for human, and then you're going to obtain a summary of the gene. So in here on the side, you can see search filtering options. This will just help you narrow down what you want to see in the database. But first, for each gene, you have basic gene information. So in here, you can see similar information to what you are able to find in UniProt. So protein, protein function, chromosomal location, and in which diseases is the gene involved in. If you scroll down, you can see a prediction of for which cells is this gene a marker, and you have here the evidence number. You can actually click on this number and see the publications that have named this gene as a marker for the specific cell type. So according to this table, CD4 is a marker for regulatory T cell. If you continue to go down, you also have information on the pathways that the gene is involved in from both the reactome pathways and wiki pathways. And finally, if drugs have been developed that target the gene you search, you can see information on these drugs in this table here. If I scroll down, for every gene, you're also able to query its expression in bulk RNA-seq. We have several datasets that can be searched to, CCLL, GTX, CCGA, for human, but we also have mouse databases such as Tismo, Vivo, and in vitro data. So this allows you to see, for example, in this cancer database, where is CD4 being highly expressed, and you can see, for example, it is expressed in leukemia samples. To move forward down, we have database-wide distribution. So you can see in which cell types is CD4 being expressed in the datasets available in SynEcoSys. So you have the top 20 cell types, and then you can see in which tissues are these cell types from. Finally, if you scroll down, you get to see the publications that have expression of CD4. And you can click on view the publication or save it for a later use. So this will be then can be found in your account under My Datasets. So today I chose to, just a second. I chose to look at Human Brain Vascular Atlas for Alzheimer's disease to give you an example of how to explore the data and an analysis part of SynEcoSys. So every time you open a publication, there's always paper information. This tells you how many cells, samples, the species, where the samples from, the publication that they originate, as well as the platform they were generated with. Below this, you have a small summary of the paper divided into research background, experimental design, and results and significance. And then you can navigate to the data and analysis part. So this starts by showing you a UMAP representation of your data. So in the UMAP, every dot represents one cell, and they are color based on cell types. And the cell types are aggregated based on the closeness of the gene expression profile. You can always highlight the publication by clicking this square here on the left. And you saw that the publication has been highlighted. And you can also hide this population by clicking on the blue square. We can call this UMAP by any information available in the dataset. For example, let's do disease subtype, and you'll be able to see the cell's color by the origin of the sample. Is it from an Alzheimer's disease sample or a healthy brain? You can then go back to the cell types. And I like to note that these plots can be all customizable. So you just click on the customize button, you get to increase the size of the dots, decrease it again, change the transparency of your dots as well. If you like to have the labels on the cluster, you just click on it. And you can also just easily change the color of the clusters. So you click on any of them, just select a different color, click on apply, and you can see that the cluster color was changed. And then you just save it. And then after you save your plot, you can download it in several different formats. So the next level of information that you have is a list of differential expressed genes. So the table shows you the top differential expressed genes for every cell type. And it's easy to navigate through the different cell types in this drop-down menu here. You can also just type any gene in this bar that you're interested in to see if it's highly expressed in a specific population. Again, all of the results seen in SynEcoSys are easily downloadable. Moving down, you can see this Explore tab, and these are the different types of analysis that we can do. And today I want just to show you an example of exploring gene expression. So the authors of this paper describe two different populations of fibroblasts, one called meningeal fibroblasts and another one called perivascular. The difference in this expression of these fibroblasts is on the receptors transporters that are expressed in their membranes. And we can try to find the same populations using SynEcoSys. So you can just type how many genes you want or upload a gene set and then click on the search function, and this will generate expression plots per each gene. So we always will be able to see expression per cluster or expression per sample. So for example, it might be that a gene is only highly expressed in one of your samples, and it's important to explore this. So as I mentioned, we want to focus on the fibroblasts to try to distinguish two populations. You can always just zoom in on your UMAP just by selecting. So the first gene that I searched for is only expressed in a small population of fibroblasts, and this should be the meningeal fibroblasts again. We can search the second gene, which should be a marker for the same population. And yeah. Again, you can see that it only labels the same small subpopulation of fibroblasts, and now we can try to look for markers of the other population, the perivascular fibroblasts, and see if they label the same population or if we are able to distinguish these two types of cells. And in here, you can see that the corner cells in here are not labeled anymore, but the other ones are. So it's very easy to find different populations if you know the markers you're looking for. Underneath, you can see a violent plot show you the expression per cell type. So moving down, again, you have the expression per sample. You can navigate through the different samples quite easily by using our drop-down menus. So now I want to show you also that we have a cellular composition. So this plot shows the average cell proportion of each cell type. But as you probably know, between samples, the percentage of a specific cell type can vary significantly. And you can look at this by selecting the specific sample from the drop down below. So for example, the largest percentage of cells in this sample number 1 is endothelial cells. But if you look at sample number 2, is oligodendrocytes. So it's important to keep in mind that the ratio of cell types will vary significantly between your samples, especially when you're considering clinical patient samples. If you move down, we can, for example, look if the conditions impact the ratios of different cells. So we can look at the ratio of different cell types between normal and Alzheimer's disease and see if there's any changes. And you can clearly see that with Alzheimer's samples, there is a decrease of endothelial cells and of neural cells, but you can also see an increase of oligodendrocytes. And then you can try to explore why do you see this difference in the cell types? What is the transcriptome changes that might be impacting this? So now we can move to differential expression. And when you click on differential expression, a heat map will be automatically generating, show you the top 3 enriched genes per cell type, as you can see here. So astrocytes, and you have the top 3 genes, plexus cells, and so on. Below, you can select groups to compare. And I decided to compare neural cells expression between healthy cells, healthy samples, and the same population with Alzheimer's samples. But you can select as many groups as you want from the metadata. Then you click on Run. And because this analysis might take a while, depending on how big the dataset is, you'll receive a notification when the analysis is done. So the result is that you get 2 tabs, one showing you the enriched genes in the first group, and the other one showing you the enriched genes in the second group that you selected. You again can search the table by typing the gene and also download this list. Moving down, you have the top 10 differentially expressed genes, and just by selecting them, you get to save them in the gene list. You have already a gene list here. You can just add them by clicking plus or remove them by clicking minus. And then this list will be saved, and you'll always be able to access it when you're trying to run this differential gene expression analysis. The selected genes can then be visualized in the same way that I showed you before. You can see the expression in both the feature plots and the violin plots. You can also use these differential expressed genes to do pathway enrichment analysis. So it's the same comparison. And there's 3 available databases that we have: Gene Ontology, Reactome, and Wikipathway. And you can see here the graph. So you have a bar graph showing you the different pathways and how they are enriched in your comparison, and also a dot plot here. So there's different ways of visualizing this information, and you can again customize these plots and download them. If we can move to the next section of data analysis, we look at cell interactions. So we use the software CellPhoneDB. So CellPhoneDB has a database of receptor and ligand pairs and how they interact, and you can search for this interaction in your cells. So it looks if your cell population number 1 has a receptor or a ligand that can interact with the receptors and ligands expressed in cell type B. So you can select which samples you want to use for this analysis and which cell types. And you can just easily select cell types and then again, click on Run. Every analysis that you do is going to be saved in this drop-down menu, and you can always come back to it. You don't have to rerun it again every time you use SynEcoSys. So let's look at an example that I've already done. And you can see how these cells interact with each other, and the thickness of the line, the thicker the line, the stronger the potential communication between the cell types. You also get to see the top 30 interacting pairs, but there are a lot of output to this CellPhoneDB that can be easily downloaded in this button here. If you want to look at a specific interaction and not at all interactions available, you can either upload a gene list, type the genes here, or select from our drop-down list. So let's, for example, select fibroblast growth factor. You can select which cells you want to consider for this comparison, and then click on plot. So the result will be a table that has every potential pair of interacting cells, so choroid plexus and endothelial cells. It shows you which interacting pairs are in the list that you just selected here. And then if it's secreted, the receptor, and you have the p-values as well. If you move then down, you have a plot summarizing this information. And you can, for example, see that there's a stronger interaction using the fibroblast growth factor between endothelial cells and neural cells. The next available option is trajectory analysis. So for this, we use Monaco 3. Again, you get to select which cell types you want to use and which samples you want to use for this analysis. So you can click on run, and again, all of the analysis you run are going to be saved here. And then you get this trajectory predicted by the software. So it predicts a change in the transcriptome of your cells. It might be differentiation, it might be response to a stimuli that goes from the dark blue to the yellow. So it says that there's a change in the mural cells from here to there. If you move down, and you can go to the find variable genes and find which genes are responsible for this progression. And you can then try to understand what is the biological relevance of this. I just want to take the time today to also show you how easy it is to initiate your own analysis. So you go to the data analysis portal, and you can first import your files. We will, SynEcoSys accepts a wide range of files, either from every platform, 10x, Singularron, BD, Rhapsody, and you can see the file descriptions in this bar here. They can be imported and then used for analysis. So to initiate the analysis, you first select your uploaded file. I have one here. And then you can add any metadata you want per sample. So let's see, we were talking about human Alzheimer's. You can, for example, add the age of the patient. And then this information that you add here per sample you'll be able to color your UMAP plots or interrogate differential gene expression by either of these columns that you add. Now you have the section for automatic cell type annotation. So we use cell ID, and we have our own cell marker list that enables you to, with high confidence, annotate your cell types. It is also important to name which species your data is from. And again, SynEcoSys is focused on human, mouse, and non-human primate data. And you also need to know where is the tissue of your sample. And this is important because the same gene might be a marker for cell type A in one tissue and cell type B in a different tissue. So referencing the tissue is also very important. And then we have cell filtering options. We have our recommended standard ones that are good to start with, but if you know that there is some specificity from your specific sample, you can always just change, for example, the percentage of mitochondrial reads just by clicking on it. You can also try to remove doublets and contaminations or cell cycle effects, and you just submit your analysis. Again, you get a notification when your analysis is concluded, and you just go again to data analysis portal, and you'll find your analysis concluded. I'm just going to take an example of that I did with lung cancer samples. You can click on view results, and it's very similar to what I showed you before. So you first see the UMAP with annotated cell types. These cell types can be found in this table. So if you don't agree with automated annotation, there's a possibility for you to do custom annotation. And we don't just do this for subtypes, but also for subtypes. For example, I already did it for T cells. So you can take your T cell cluster and try to differentiate the different types of T cells present in your sample. So this is just a very quick overview of what SynEcoSys is able to do. It can always do more than what I showed, and you can take this time to explore it as well. But I just like to quickly summarize it. So SynEcoSys is a data repository. This is the numbers we have currently. We have over 45 million cells, and we cover over 250 diseases. The database is updated on a weekly basis, so these numbers are constantly changing. For diseases for which we found a lot of high-quality publications, we create core datasets. So in these, we integrate multiple datasets to have a larger cohort that you can easily interrogate for your sample of interest. Our data analysis platform currently covers most of the single-cell RNA-seq analysis, but it's also something that's being constantly updated. So I want to mention that if you're interested, in either SynEcoSys or single-cell sequencing kits. If you use our kits or our service, you are able to use SynEcoSys for free to analyze the data generated. And if you need more information, you can always contact our info email, or if you want more information, you can contact my colleague, Benny. You can see here his email, or look at our website, or follow us on LinkedIn. I just like to take this opportunity to also mention that Single-On is offering a single-cell analysis bootcamp. So it's a 2-month course with 8 hours per session, 1 session per week, that will run through the entire single-cell analysis pipeline and make you able to do your analysis independently. So with this, I'd like to thank you for taking the time to listen to my talk, and I'll take any questions that you have now.
Okay, thank you, Anna. So I see that there are 9 questions. So before we proceed to the Q&A part, I'd like to do a quick reminder. Do remember to fill in the post-webinar feedback form as well. So the link is actually sent in the chat. Okay, so let's go through the questions. We'll answer as many as we can. So there is a question on from an anonymous attendee. Is batch correction applied in SynEcoSys across the integrated samples or studies?
Yes, so we do apply batch correction and usually use Harmony to do this. So if I can just show you something else very quickly. So we have these resources for SynEcoSys, so you can go through the documentation and see in detail every software that we use for every step of the analysis and what are the parameters we also use to do it.
Okay, thank you, Anna. So the next question is there a guide for this system?
Yes, so you can see also in the resources, there's a guidebook, and the guidebook shows you step by step with print screens how to use every single part of this. In our YouTube page, you can also find short videos, like 2 to 3 minute videos on how to use every section of the analysis as well.
Okay. Next question, is it possible to integrate different data sets? So for example, checking a gene across data sets from multiple papers on brain tumors.
So currently no, but this is an update that will come not so late in this year as well. So you will be able to integrate different data sets.
Okay. The next question, is there a way to upload data into the system?
Yes, so as I show, you can go to a data analysis portal, initiate data, import files. You just drag any file into here and it will be uploaded. If you are a customer and you like to ask us to add available public data, you can also always submit a request and we will add that data to the database for you.
Okay, thank you, Anna. Okay, the next question has so far the most slides I see. So how much do you charge? Is there a subscription fee for this?
So there is a subscription fee, it's a monthly fee. And for more details, it will really be better if you contact my colleague, Benny. I can again share his contact. And he'll be able to ask you in. We need to share again.
Yeah, we can see your screen. Yeah.
I'll just leave his contact up so you can take it. Yeah.
Okay. The next question. Hi, may I know what batch correction method can I use with SynEcoSys?
So the batch correction method is Harmony, as I mentioned before, and you can also find the reference in the documentation page.
Okay. The next one, what would I have to do to pull data from the database and combine with my own data to do trajectory analysis with different time points?
So for every data set available, you can see okay, I will show you again because it's always easy to show. So let me, okay, let's go to any publication. So you can see here the download portal. So you always can request to download these data sets and then upload it to combine with your own data in the same way that I showed you before.
Okay, we have a question from Ling Wu. Do you indicate which analysis method you're using for these analyses in your system?
Yes, so for the sum of them is written in the part. So for cell interaction analysis, we showed you that is cell phone DB. For trajectory is Monaco 3. But again, in the resources, in the documentation, you can see what is used for every single step of the analysis.
Okay, there is a question that's asking about trial account. So do you have a trial account that we can use?
So if you go to the SynEcoSys page, synecosis.com, you can apply for a 5-day trial. It's not full access, but it's enough for you to explore a few data sets.
Okay, over here I see the last question from Yi Tian. Is it possible to analyze TCR or BCR abundance and diversity?
So we already have some data sets, but this is something that is coming with the next upgrade of the software that's coming later this year as well.
Okay, thank you so much for all the questions. So if you have any other questions, do quickly send it in, and now over here will answer and clarify with you. Maybe Anna, could you flash the contact emails again?
Yeah, I will add the contact. So yeah, if you have
Yes.
any more questions, you can find it all in our website, and we also have regular updates in our LinkedIn page.
So I don't see any questions in the Q&A chat box. So thank you everyone for attending today's webinar. If you have further questions about the system or you want to find out more about this, so do feel free to reach out to them. The email is on the screen. So thank you everyone for attending today's webinar. We'll see you in the next one.
Thank you.
Thank you.